Osmoregulation is the active regulation of the osmotic pressure of an organism's body fluids to maintain the homeostasis of the organism's water content. That is, it maintains the fluid balance and the concentration of electrolytes, salts in solution, to keep the fluids from becoming too diluted or too concentrated. Osmotic pressure is a measure of the tendency of water to move into one solution from another by osmosis. The higher the osmotic pressure of a solution, the more water tends to move into it. Pressure must be exerted on the hypertonic side of a selectively permeable membrane to prevent diffusion of water by osmosis from the side containing pure water. Organisms in aquatic and terrestrial environments must maintain the right concentration of solutes and amount of water in their body fluids. This involves excretion or getting rid of metabolic nitrogenous wastes and other substances such as hormones that would be toxic if allowed to accumulate in the blood, through organs such as the skin and the kidneys. Two major types of osmoregulation are osmoconformers and osmoregulators. Osmoconformers match their body osmolarity to their environment actively or passively. Most marine invertebrates are osmoconformers, although their ionic composition may be different from that of seawater. Osmoregulators tightly regulate their body osmolarity, maintaining constant internal conditions. They are more common in the animal kingdom. Osmoregulators actively control salt concentrations despite the salt concentrations in the environment. An example is a freshwater fish. The gills actively uptake salt from the environment by the use of mitochondria-rich cells. Water will diffuse into the fish, so it excretes a very hypotonic urine to expel all the excess water. A marine fish has an internal osmotic concentration lower than that of the surrounding sea water, so it tends to lose water and gain salt. It actively excretes salt out from the gills. Most fish are stenohyline, which means they are restricted to either salt or fresh water and cannot survive in water with a different salt concentration than they are adapted to. However, some fish show a tremendous ability to effectively osmoregulate across a broad range of salinities. Fish with these abilities are known as euryhaline species, or the flounder. Flounder have been observed to inhabit two utterly disparate environments, marine and freshwater, and it is inherent to adapt to both by bringing in behavioral and physiological modifications. Some marine fish, like sharks, have adopted a different, efficient mechanism to conserve water, and that is osmoregulation. They retain urea in their blood in relatively higher concentration. Urea damages living tissues, so to cope with this problem, some fish retain trimethylamine oxide. This provides a better solution to urea's toxicity. Sharks, having slightly higher solute concentration, do not drink water like freshwater fish. While there are no specific osmoregulatory organs in higher plants, the stomata are important in regulating water loss through evapotranspiration, and on the cellular level, the vacuole is crucial in regulating the concentration of solutes in the cytoplasm. Strong winds, low humidity, and high temperatures all increase evapotranspiration from leaves. Abscisic acid is an important hormone in helping plants to conserve water. It causes stomata to close and stimulates root growth so that more water can be absorbed. Plants share with animals the problems of obtaining water But unlike in animals, the loss of water in plants is crucial to create a driving force to move nutrients from the soil to tissues. Certain plants have evolved methods of water conservation. Xerophytes are plants that can survive in dry habitats, such as deserts, and are able to withstand prolonged periods of water shortage. Succulent plants such as the cacti store water in the vacuoles of large parenchyma tissues. Other plants have leaf modifications to reduce water loss, such as needle-shaped leaves, sunken stomata, and thick waxy cuticles as in the pine. The sand dune marram grass has rolled leaves with stomata on the inner surface. Hydrophytes are plants in water habitats. They mostly grow in water or in wet or damp places. In these plants, the water absorption occurs through the whole surface of the plant. For example, the water lily. Halophytes are plants living in marshy areas close to sea. They have to absorb water from such a soil which has higher salt concentration and therefore lower water potential, higher osmotic pressure. Halophytes cope with the situation by activating salt in their roots. As a consequence, the cells in the roots develop lower water potential, which brings in water by osmosis. The excess salt can be stored in cells or excreted out from salt glands for leaves. The salt secreted by some species help them to trap water by pores from the air which is absorbed in liquid by leaf cells. Therefore, this is another way of obtaining additional water from air. For example, glasswork and cordgrass. Mesophytes are plants living in the land of temperate zone which ground when watered soil. They can easily compensate the water loss by transpiration through absorbing water from the soil. To prevent excessive transpiration, they have developed a waterproof external covering called cuticle. Kidneys play a very large role in human osmoregulation by regulating the amount of water reabsorbed from glomerular filtrate in kidney tubules, which is controlled by hormones such as antidiuretic, aldosterone, and angiotensin II. For example, a decrease in water potential is detected by osmoreceptors and hypothalamus, which stimulates and release from pituitary gland to increase the permeability of the wall of the collecting ducts in the kidney. Therefore, a large portion of water is reabsorbed from fluid to prevent a fair proportion of the water from being extracted. A major way animals have evolved to osmoregulate is by controlling the amount of water lost through the excretory system. Amoeba makes use of contractile vacuoles to collect excretory waste such as ammonia from the intercellular fluid by diffusion and active transport. As osmotic action pushes water from the environment into the cytoplasm, the vacuole moves to the surface and disposes the content in the environment. Bacteria respond to osmotic stress by rapidly accumulating electrolytes or small organic solutes via transporters whose activities are stimulated by increases in osmolality. The bacteria may also turn on genes and coating transporters of osmoles and enzymes that synthesize osmoprotectants.